Since the discovery of DNA in 1950s and the invention of methods to read the DNA sequences, our understanding of life has grown at a tremendous pace. DNA and protein sequences have taken a central place in all biological and medical investigations. One of the key tasks in utilizing sequences is to compare them and assess their similarity. In this lecture, we will first discuss the criteria used for evaluating similarity of sequences. Although there are other ways of quantifying sequence similarity, I will mostly focus on sequence alignment. And I may say similarity when I should really be saying alignment, and vice versa. In sequence alignment, we lay out sequences side by side, matching them character by character, so that the parts that are similar can be identified, and the corresponding characters can be scored using a substitution matrix. After we review sequence similarity, we will discuss the algorithms used to perform different types of sequence alignments. Sequence alignment helps answer a large number of questions about genes and organisms. We align sequences to assess if they are related, if they have evolved from the same ancestral sequence. Similarity helps us annotate new genes and discover their roles toward the functioning of the cell and the organism. We can identify the corresponding residues between sequences to pinpoint exactly what mutation is responsible for a genetic disease. Microarray is a method used to quantify the expression levels of genes at a genomic scale. The probes on a microarray chip are designed using sequence similarity to ensure that they can uniquely measure the expression of a particular gene. Shotgun assembly is a method used for constructing the entire genome sequence of an organism using small chunks of sequences. Putting together these small puzzle pieces makes use of sequence similarity to put them together, with or without a reference picture we can look at. Using sequence similarity, we can find out if a gene is present in other species and how conserved it is across different species. And by comparing related genes across different species, we can identify residues or motifs that are important for the function of that gene. When we compare sequences, we are implicitly trying to uncover the evolutionary history of those sequences. Let's review some related terms used in the evolution of biological things. Biological structures may be similar due to two evolutionary mechanisms: divergent evolution and convergent evolution. Things may be similar because they have evolved, they have diverged starting from the same ancestral structure, such as the formation of the limbs in the vertebrates. The term we use to describe structures resulting from divergent evolution is homologous. We sometimes say things are homologous because they are similar, especially when we are dealing with their genetic sequences, and use the terms homologous and similar interchangeably. But we have to keep in mind that things can be similar due to other reasons as well, such as in convergent evolution, which is also known as parallel evolution, where different species can acquire the same sequence, structure, or function independently. We use the term analogous to describe the relationship between such structures. Some of the well-known examples are the wings on bats, birds, and butterflies, the eyes of the vertebrates and cephalopods, which include the squid and octopus. These structures have not evolved from a common wing structure or from a common eye organ, but rather they were independently discovered at different branches of the evolution. There are several types of evolutionary events that give rise to homology. The genes that are similar because of a speciation event are called orthologs. When we have a speciation event, each species has a slightly different version of the gene. These orthologous versions would keep changing ever so slightly from generation to generation. If the two species are closely related, we can compare the sequences of the orthologs and observe that they are similar. If the speciation happened a long time ago, we would call these genes distant homologs, and the similarity between the sequences may not be as obvious. Sometimes we have a gene duplication event, where within the same species, we have multiple copies of a gene. These genes are called to be paralogs. The tree structure on this slide demonstrates the evolutionary history of a globin gene. Globin is a protein that can bind and transport oxygen. Such as in red blood cells, globin binds oxygen in the lungs and carries and releases it throughout the body. The globin gene is widely conserved, meaning many different types of species have it. It is important to note that the information we have is primarily the sequences of the organisms available to us at this moment in history. We don't really have access to the genetic sequences of fossils that date far back enough to help us in discerning the evolutionary history. So we have to infer the history from the sequences that are available to us now. Construction of such an evolutionary tree is the topic of phylogenetics. At the root of this tree, there was a speciation event, separating lamprey from the other species listed in this figure. Lamprey is a jawless fish that is sometimes incorrectly called a lamprey eel. Lamprey has a globin gene, which is orthologous to the globins in these other species. The lamprey globin and the globins in these other species are related to each other because of a speciation event. After this speciation event, there was a gene duplication, giving rise to two copies of the globin within each of these species, the alpha globin and the beta globin. We know that the gene duplication happened before the subsequent speciation events, because the frog alpha globin is more similar to mammal alpha globin than it is to frog beta globin. Frequently, after a gene duplication event, one of the copies of the gene mutates into a non-functional gene, also called a pseudogene. Since the organism has another copy, it can afford to lose the extra copy. Sometimes this extra copy can also evolve to accomplish a different function. Another type of evolutionary event is when a genetic material is transferred from one species to another. We call this horizontal gene transfer, and the resulting genes are called xenologs. The classic example of horizontal gene transfer is the spread of antibiotic resistance in bacteria. Antibiotic resistance genes are transferred from one bacteria to another by plasmids, which are small circular chromosomes, and by bacteriophages, which are viruses that infect bacteria. Horizontal gene transfer does not just happen in bacteria. It is estimated that about 100 genes in humans have resulted from horizontal gene transfer. Gene duplication and horizontal gene transfer events can complicate the study of species. In a hypothetical scenario, let's say the frogs and mammals lost their beta globins, and the snake lost its alpha globin. Then we would have a single globin in each of these species, and we would not have known about the gene duplication event, and we would just call them all globins. Because the alpha globins are more similar to each other, based on the analysis of the globin sequences, we may incorrectly claim that frogs and mammals are more closely related to each other than mammals and snakes. It is important to note here that the gene tree may not accurately reflect the species tree, because of evolutionary events other than speciation can give rise to a particular gene tree. Using a single gene is akin to using a single feature, such as living in the sea, to classify animals. Dolphins may be similar to other fish in that they also live in the sea, but the species tree would put dolphins and humans under the same branch and the fish on another branch. The tree of life, the tree of species, is typically constructed from very carefully selected genes where duplication events would not confuse the analysis. And we would also use evidence from multiple genes to construct a more robust tree. Genetic sequences change from one generation to the next because of mutation events. Substitutions are when one type of nucleotide is replaced with another type of nucleotide. Substitutions can happen during replication of the DNA. DNA replication is a high-fidelity process giving a near-exact copy of the DNA. The replication machinery does make mistakes. Some of these mistakes are corrected by DNA repair mechanisms. When they are not corrected, the daughter cells receive a copy that is not identical to the DNA in the parent cell. In humans, there is about 1 in a billion nucleotides error when the DNA is replicated. For viruses, whose survival depends on creating many variants, the error rate is higher. Mutations of the DNA can also happen within a cell when it is not replicating, such as due to chemical agents. Reactive oxygen species that are byproducts of chemical reactions that go on in the cell, and ultraviolet light can cause damage and mutations in the DNA. Insertion or deletion of the DNA segments can also happen during DNA replication or due to other mechanisms, such as transposable elements or transposons, which are sequences of DNA that jump from one location to another. Viruses can also integrate their genetic material into the host DNA, which can be considered as an insertion event. Duplication of genetic material can also happen during replication, and it can happen at different scales. An entire gene can be duplicated, resulting in multiple copies of a gene. A section of a gene can be duplicated or moved, resulting in domain duplication and exon shuffling. A chromosome can be duplicated in its entirety or partially, which are called polysomy or partial polysomy. Some human diseases are caused by polysomy, such as in Down syndrome, the affected individuals have 3 copies of chromosome 21. Polysomy is caused by incorrect segregation of the replicated chromosomes into daughter cells during cell division. When there are multiple copies of the entire genome, we call that polyploidy. Most eukaryotes are diploid, meaning they have 2 paired homologous sets of chromosomes, one inherited from each parent. Polyploidy is having more than 2 sets of chromosomes and is typically found in plants. Now that we have reviewed the types of mechanisms that cause changes in the genetic material, let's now see how we can compare sequences and sort of play detective to figure out if they have evolved from the same ancestor gene and which of their nucleotides have been substituted, inserted, or deleted. Let's say we have these 3 sequences that we have obtained from frogs, humans, and snakes. Can we say that sequence is more similar to this is a sequence than it is to the frequence? For short sequences, we may be able to recognize their similarity just by looking at them. But what if these sequences were 1,000, 1,000,000, or 1,000,000,000 characters long? Can we come up with a systematic method for comparing them? There are different ways of comparing sequences to identify their similarity. For example, in one approach, we can go through each sequence and extract the frequency of each character. In this way, we are summarizing a sequence, no matter how long it is, to an array of 20 numbers, assuming we are dealing with protein sequences and we have 20 types of amino acids. Once represented in this way, we can use any one of the distance or similarity measures, such as Euclidean distance or correlation, to quantify how different or how similar any two sequences are. The frequency representation is an oversimplification. If the sequences have the same composition of characters, it ignores the particular arrangement of the characters and would declare the two sequences to be similar. Ideally, we need a method that examines not only the character composition of the sequences, but also the arrangement of these characters. In the sequence alignment, we put sequences side by side, or one on top of the other to be exact, and line up their characters. This lining up of their characters implicitly assumes that the corresponding characters have the same position when they mutated from an ancestral gene. Once lined up in this way, we can count the number of characters that are the same and that are different and use that information to quantify similarity. When we compare two sequences, we usually don't have access to the ancestor gene because the ancestor species has long died out. By comparing the two descendant sequences, we may be able to infer the ancestor gene. When characters of the genes we are comparing are the same, we assume that the ancestor also had those characters. This assumption is not always true, as you see from the first character here, which has been mutated into a T in both of the descendants. When characters are different, we assume that this is a result of mutations from the ancestor gene. If one sequence is shorter than the other one and we line them up, we would end up pairing characters that are not from the same origin and would incorrectly make inferences about the mutations that must have happened. Our alignment must model not only the substitution events, but also insertions and deletions. We use dashes to indicate that part of one sequence has been deleted or there was an insertion at that position in the other sequence. Given two sequences, there are obviously many different ways we can align them, based on where we place these gaps. Here you see three different alignments of the same two sequences. Each alignment gives us a different number of matches, mismatches, and gaps. Which of these alignments is correct? The question is not easy to answer. We need to come up with a metric that tells us how good each alignment is. Roughly speaking, the higher the number of matches and the lower the number of mismatches and gaps, the better the alignment. One way to put this idea into a formula is to add the number of matches and subtract the number of mismatches and gaps, giving us a score of 2 for the first alignment, 3 for the second one, and 2 for the third. So with this approach, we would say that the second alignment is the best of these three. What are some of the other ways we can evaluate an alignment? Percent identity is a commonly used intuitive number we report to talk about how similar two sequences are. It is calculated as the number of characters that are the same divided by the length of the alignment. The length of this first alignment is 16, and 11 of these positions are identical, giving us 69% sequence identity. The second alignment here is shorter because there are fewer gaps. 10 out of 15 positions are identical, giving us 67% identity. In some applications, we may divide by the length of the shorter sequence rather than the length of the alignment. With this alternative definition, for this first alignment, we would get 11 divided by 12 because the length of the shorter sequence is 12, which results in 92%. Something as trivial and conventional as percent identity can have wildly different values depending on how you choose to calculate it. If you are reporting percent identity in your own results, make sure you specify how you calculated it. Scoring matrix is an extension of percent identity where instead of having all pairings scored equally, we give higher scores to some pairs of characters and lower scores to others. We'll discuss scoring matrices in more detail later. A dot plot is a qualitative visual method of evaluating alignment. To create a dot plot, one of the sequences is written on the columns of a table, and the other sequence is written on the rows of the table. A dot is placed in the table wherever the pairs of characters from the two sequences are identical. For large sequences, the dot plot can look pretty messy. A window filter is applied to remove some of that noise and obtain a figure that is easier to interpret. Here's an example where we have an unfiltered dot plot and we apply a filter using a 5x5 window centered around each of the cells to decide whether we keep that dot or filter it out. Looking at the diagonal of this window, we have dots in at least 60% of the cells, so we keep that central dot. In this second window, we only have 2 out of 5 dots, which doesn't pass our 60% threshold. So we filter out the central dot for that window and not show it in the cleaner filtered dot plot. We repeat this for every dot in the dot plot and we either keep them or remove them. The window size and the match threshold can be adjusted based on the size of the matching segments you want to end up with. A window size of 3 with exact matches is a common threshold, giving us only stretches of subsequences from the two sequences that are at least 3 characters long. Looking at a dot plot of two sequences, we can identify certain local patterns. Let me call this vertical sequence P and the horizontal sequence Q. A segment of matching characters between the two sequences would appear as a diagonal line. A stretch of mismatches or substitutions would appear where a diagonal line is interrupted by a stretch of blank space. An insertion is where a diagonal line ends somewhere and starts again down below, meaning that P has a segment that Q is missing. A deletion can be detected likewise, where now Q has a segment that P is missing. An inversion is where we have a segment of characters that are identical between the two sequences, but these characters are arranged in one direction in P and in the opposite direction in Q. We can create a dot plot of a sequence with itself to identify any repeating patterns within that sequence. In a self-dot plot, we would of course have the main diagonal line because every character is equal to itself. The repeating patterns will appear as line segments other than the main diagonal. Inverted repeats would also be detected as line segments that are slanted in the other way. If we get an inverted repeat that crosses the main diagonal, that would tell us that there is a palindromic pattern, meaning you get the same sequence if you were to read it backward. A block of short lines indicates the presence of a low complexity region, which is formed by multiple repetitions of a short sequence pattern. Percent identity and dot plots are based on exact matches. Percent identity assumes that aligning a T with a T is as good as aligning an H with an H, and that aligning an A with an I is as equally bad as aligning a T with an S. For DNA sequences where we have 4 characters, ACGT, this equal contribution of different pairings is acceptable. But when we examine substitutions that do happen in protein sequences, we observe that some pairings are more frequent and favorable than others due to biochemical properties of amino acids. In this alignment, aligning A and I is actually not such a bad thing because they are both hydrophobic amino acids. Aligning T and S is also not so bad because they are both polar amino acids with a hydroxyl group on their side chain. A and I are more interchangeable than, say, A and W. We capture the favorability of pairing different amino acids in an alignment in what we call a scoring matrix. A scoring matrix would, for example, say aligning an A with an I has a small penalty of -1, but aligning an A with a W, because they are less compatible, should have a larger penalty of -3. Blossom 62 is one of the popular substitution matrices we use for scoring alignment of amino acids. I will describe how we make use of this matrix first, and then explain how they produce this matrix. When we look at an alignment, for every matching or mismatching character pair, we can now look at the substitution matrix and use the score for that pair. For example, aligning a T with a T is worth 5 points. Aligning an H with an H is worth 8 points. Aligning an A with an I is worth -1 points, and so on. Adding up all these points gives us the total alignment score, in this case 52. A substitution matrix is a symmetric matrix, so showing only half of the matrix is enough. The other half will contain the same values. Notice that the diagonal values are not the same. Aligning a tryptophan amino acid, which is shown with a W, with another tryptophan is worth 11 points, whereas aligning a valine with a valine is worth only 4 points. The tryptophan amino acid prefers to align with itself more than valine prefers to align with itself. And this is reflected with the large self-pairing score of tryptophan compared to self-pairing score of valine. Let's now see how the Blossom matrix, which stands for block substitution matrix, was produced. What they did was take a collection of manually created alignment of blocks of sequences. Here an example block of sequences is shown. If we use a sufficiently large number of such blocks, we can hope that the frequency of mutations in this large dataset would reflect the frequency of mutations that happen in nature. The idea is to use this blocks dataset to quantify, for any pair of amino acids, how associated or favorable they are. For each block, we first count the frequency of observing each of the different pairs of amino acids. We also count the background frequencies of each amino acid. The odds ratio is defined as the frequency of observing an event divided by the frequency at which we expect it to happen by random. In other words, it is defined as observation divided by expectation. Here we divide the frequency of observing two amino acids together, I and J, divided by the multiplication of their background frequencies, which gives us the expected frequency that they would be observed together by chance. Blossom uses the logarithm of this ratio for numerical convenience. This allows us to add up the scores rather than to multiply them. Blossom also rounds the resulting scores, so we end up with integers. The idea behind rounding is that computers are faster in performing integer operations than they are with real numbers. Let's walk through a small example. Consider the following block of 4 sequences, each with 4 amino acids. The background frequency of A is 7/16 because we have 7 A's out of 16 total characters. Similarly, the background frequency of D is 5/16. The number of times that an A is replaced with an A is 12. Let's count. In the first column, there are no pairs of A's. In the second column, there are no pairs of A's. In the third column, there's 1, 2, 3, 4, 5, and 6 pairs. But we have to remember to use each A as a source, so going the other way will give us another 6, giving us a total of 12 times. The number of times an A is replaced with a D is 5. Let's count those. In the first column we have 2. In the second column we have no pairing of A and D. In the third column we have no pairing of A and D. And in the fourth column we have 1, 2, 3, giving us a total of 5 pairs of AD. To find the frequency of AA, we'll divide the number of times we see AA divided by the total number of character pairings, which is 48. The log odds score of AA is calculated as 2 times log 2 of PAA divided by the multiplication of background frequencies of A and A. The log odds score of AD is calculated as 2 times log 2 of PAD divided by QA times QD. Because of the way the BLOSUM matrix is generated, using BLOSUM to score an alignment gives us a nice probabilistic interpretation of the alignment. In this example, when we add up the BLOSUM scores of the amino acid pairs in this alignment, we get a total score of 52. Remember that adding these scores, which are on a logarithmic scale, is equivalent to multiplying the log odds ratios of observing these amino acid pairs. A score of 52 is equal to a log odds ratio of 2 to the power 26. Therefore, this alignment is 2 to the power 26 times more likely to be seen in a real alignment than expected from a random alignment. A few slides back, I showed the BLOSUM 62 matrix. What does the 62 mean? When they generated the BLOSUM, they clustered the sequences at different percent identity levels. The clustering of the sequences into different groups affects how the character pairs are counted. BLOSUM 80, which is generated by clustering highly similar sequences, would reflect the types of substitutions that are present in highly similar sequences. BLOSUM 45 is generated by clustering highly divergent sequences and would reflect the types of substitutions that are present in remotely related sequences. BLOSUM 62 is sort of an average case and is the most commonly used matrix in many bioinformatics applications. If you have a specialized project that deals with highly similar or highly divergent sequences, you may wish to use a different matrix. There are a large number of other types of substitution matrices available. Some of them have been shown to give higher quality alignments than BLOSUM. Despite worse accuracy, BLOSUM 62 has remained the default matrix in most applications. PAM is another matrix that, like BLOSUM, starts with a dataset of pre-aligned sequences. But instead of looking at all pairs of characters in a column, it first constructs an evolutionary tree to identify more accurately which specific mutations have occurred. Using these mutation counts, it also uses log odds ratio to calculate a score. Substitution matrices provide a well-formalized method to score pairings of one character with another, except for the gap character, which is usually not considered as part of a substitution matrix. The specific value of gap penalties are typically determined empirically through trial and error to find a value that best replicates manually curated alignments. There are two commonly used gap penalty models. In the linear gap penalty, which is more straightforward, each gap character is penalized by a gap penalty value. A typical value for the gap penalty is 8. When we look at each of these alignments, we see that they both have 3 gap characters. So for both of these alignments, the contribution of the gap penalty toward the total alignment score is minus 3 times 8, which is equal to minus 24. In some applications, you may need to pay attention to how the gap penalty is specified. You need to make sure that the contribution of the gap penalty toward the overall alignment score is always negative. In this example, I may tell you that the gap penalty is 8 or I may tell you that the gap penalty is -8. Regardless of whether I gave you a positive or negative number, it is your job to negate it and get -24 before you add it to the overall alignment score. Mechanistically, insertion and deletion events happen not character by character, but segment by segment. So a single insertion event can account for a stretch of 3 gaps, whereas 3 different insertion events would have to have happened to produce the second alignment. It would make sense to not penalize the first alignment as much as the second one, or adjust our scoring model so that the first alignment has a higher total score than the second one. An approach to accomplish this is to use a fine gap penalty. In a fine gap penalty model, we distinguish between a gap opening character and a gap extension character. A gap opening is the first character in a stretch of gaps. In this first alignment, there's a single stretch of gaps, and the first gap character is the gap opening. The remaining gap characters are gap extension characters. Let's say gap opening penalty is 8 and gap extension penalty is 3. For the first alignment having 1 gap opening and 2 gap extension, we would end up having -1 times 8 minus 2 times 6, which is -14. In the second alignment, there are 3 gap segments, each of them composed of a single gap opening. So for the second alignment, the gap penalty is -3 times gap opening penalty, which is 8, minus 0 times gap extension penalty 3, which results in -24. The first alignment, because it has fewer gap openings, has a smaller gap penalty, and the overall alignment score will be higher. When we are comparing 2 genetic or protein sequences, we may want to know how they align in their entirety, which we call global alignment, or whether they share a smaller, highly similar segment, which we call local alignment. Local alignment is especially useful when comparing multi-domain proteins. A global alignment may mess up the alignment of these domains when it tries to align the sequences in their entirety. This concludes our discussion of sequence similarity and alignment. We are going to learn about how global and local alignments are constructed in another lecture.